produce collagen, or blood comes in touch with the activated platelet, right? Then coagulation factor number 12 is activated, or contact factor is activated, and intrinsic pathway triggers. Is that right? Plus, they say when there is injury, extrinsic pathway is also activated. Do you think there is mouse and brain of the mouse there? No. Actually, what was in the brain mouse? There was tissue factor. What we call it? Tissue factor or thromboplastin. Later on, we came to know that thromboplastin factor or tissue factor is just some lipids plus some lipoproteins, which can activate factor number 7. So they came to know if you take any human cells and mash them and crush them, they will produce some tissue juice and tissue factor. And that tissue factor is capable of activating extrinsic pathway. Of course, when there is injury here, then platelet will produce tissue factor, injured endothelial cell will produce tissue factor. So that tissue factor, right? What is it? Tissue factor and tissue factor. These tissue factors can activate which pathway? Extrinsic pathway. Now let me tell you exactly what are these intrinsic pathway and extrinsic pathway. But the most important thing which you need to remember is that either it is intrinsic pathway or extrinsic pathway, the end should be the same. Conversion of soluble fibrinogen into insoluble fibrin strand. That's it. Now let's start how it occurs. Here is your liver, and liver is producing a lot of coagulation factors releasing into circulation all the time. Right? The first event here is in intrinsic pathway. Which factor will touch? Factor number? Yes, 12. So what happens? Factor number 12 will convert into active factor number 12, where the site where platelets are activated, or endothelial cells are injured or activated, or subendothelial collagen is exposed. So all these factors have a capability to activate the Hageman factor or contact factor or coagulation factor number 12 into activated factor. So what was this? This was an enzyme. What was this? This was an enzyme which was again sleepy. And activated factor number 12 is active factor number 12. So whenever a factor number 12 reaches at the site of injury in the vascular system, factor number 12 automatically gets activated. Now this active factor number 12 will activate, yes, it will activate which factor? Factor number 11, right? And this factor number 11 will be now, yes, activated. Factor number 11 will be activated. Okay, we'll make it like this. Inactive, activated or circled. So first factor number 12 is activated. Activated factor number 12 will activate factor number 11. Factor number 11 will activate factor number, yes, will activate factor number 9. Yes, you are too good. And factor number 9 here is activated. Is that right? Now, there is something very important, right? That once factor number 9 is activated, in the presence of 9, in the presence of phospholipids and calcium and factor number 8, in the presence of all these substances, okay, I'll write it at the top. 9 in the presence of phospholipids. Of course, those phospholipid is the floor which is provided by platelets. Platelet surface is having phospholipids. So phospholipids, and in the presence of calcium and in the presence of factor 8. Factor number 9 in the presence of phospholipid, calcium and factor number 8 can activate now factor number, what is this? 10. Factor number 10 will be activated. Is that right? Now, once the factor number 10 is activated, factor number 10 will act on prothrombin. It will act on thrombin and convert that prothrombin into thrombin. And this was the real action, because this is the thrombin, right? This is the thrombin this is the superactive molecule, this is the thrombin, which will, yes, which will convert, yes, what thrombin will do? Fibrinogen, yeah, it will break down the fibrinogen into fibrin monomers. Fibrin monomers. So during all this process, look, factor number 12 activate factor number, first factor number 12 is activated, that will activate factor number 11. 11 activated, 11 will activate the factor number 9. 9 factor number activated factor number 9 in the presence of phospholipid, calcium, and what is this? Factor number 8 will activate factor number 10, and factor number 10 again in the presence of factor number 10 in the presence of calcium and phospholipid, and factor number 5 will convert prothrombin into thrombin, and thrombin will break down the fibrinogen to fibrin monomer, right? And these fibrin monomer will deposit here. What are these? As this reaction will continue again and again, right? And thrombin will keep on activating the fibrinogen to fibrin monomers, and all these reaction is occurring on the surface of platelets, and on the platelets lot of fibrin monomers are deposited. Meanwhile, thrombin also activate another substance which is called thrombin stabilizing factor, that is activated. What is this? Thrombin stabilizing factor, which is also called factor number 13. Factor number 13. So thrombin can produce the fibrin monomers. Thrombin can produce the fibrin monomer. At the same time, it activates factor number 13, which is fibrin stabilizing factor, and fibrin stabilizing factor or activated factor number 13 is an enzyme which will rush on these monomers and produce what? Cross-linking of these monomers. So all these monomers get cross-linked. Now this become a strong covalent meshwork and network of fibrin on the platelets and within the platelet plug. Within the platelet plug and on the surface of the platelet. Now this primary platelet plug has been converted into secondary platelet plug or secondary hemostatic plug. Am I clear? Meanwhile, we should not forget that tissue factors were also produced. You remember that from injured area, tissue factors were also produced. This tissue factor will convert now extrinsic pathway, factor number 7 into active 7. So factor number 7 can be activated in the presence of tissue factor, which may be coming from platelet, may be coming from injured endothelial cells or other injured tissue. An activated factor 7 can do two functions. Now activated factor 7 can also, right, can also activate factor number 9, and it can also activate factor number 10. Right, which pathway is short? Extrinsic or intrinsic? Extrinsic. So which is faster? Extrinsic. The short pathway and explosively work very rapidly. Extrinsic pathway is a fast system. Intrinsic pathway is a slow system. In this way, coagulation occurs on the surface of the platelet plug, and platelet plug convert from primary platelet plug to secondary platelet plug.